Growth and shape changes to the skull are traditionally considered to stop once one has reached maturation, or adult age. However, studies on adult primates suggest that craniofacial changes are evident. After all, a 20-year-old and a 60-year-old look very different from each other. This project aims to find out how much age affects cranial shape, as well as what may cause such changes. Our sample uses crania from wild baboons, where their ages are unknown, and since age is often related to enamel wear, we decided to use the upper third molar's wear stage as a proxy for adult age. In order to do this effectively, we scored the molars on a scale of 0 to 16. As you can see on figure 2, a younger baboon would have a molar wear score of 0 or 2 with no wear or minimal wear, and an older baboon would have a score of 13 or 16, with a lot of the enamel gone. If you look at Table 1, you can see that we use 6 subspecies of baboon under the taxon Papio, with 221 males and 126 females, making a total dataset of 347 baboons. All of the statistical analyses were done with the Geomorph package in Rstudio. If you look at Figure 1, you can see 29 of the 45 landmarks we took from each specimen with a microscribe, illustrated with the little red dots. Next, we superimposed the data, standardizing the geometric size of the specimens while leaving the shape differences. Then, we tried to mitigate the effects of allometry by putting the superimposed data through a multivariate regression analysis. Figure 3 shows these effects of allometry. Males, in blue, are generally bigger than females, which are in red. The results demonstrate that size accounts for the majority of shape differences, taking up 42% of the variation. While subtle, age does have an effect too, with 6% of the shape variation being caused by age. Therefore, we were successful in finding clear evidence for age-correlated shape changes. Next, we can start to work towards investigating what these changes actually are and what causes them. These effects of aging on cranial shape can help forensic studies, help comparisons of fossil specimens, and help us to understand the general aging process. So further work on this project in the long run might discover what causes these age-related changes. Are these changes caused by genetic issues, or could they be shaped by something else? What could the processes of these shape changes be, and why do they happen? Whatever the cause, and whatever the reason, we look forward to investigating in the near future.